Lynch. Professor, he is from Glasgow Smith Lane, GSK, Swansea University Medical School, UK. He has credit of 100 plus publications for his contribution. He has got a high H index of 33 and he has got a good awards of UK EMS Young Scientist Award in the year 1998. You can applaud. He got an UK award. Jim Perry Award in 2025 and he is a fellowship of FRC Park, Honorary Chair in Swansea University, Wales, UK. And he also past President in UK EMS and past Treasurer in EMTS, Chair in ECNGS, SAG UK. He has got a lot of administrative positions on his credit. I request Dr. Anthony M. Lynch to speak about his talk. Thank you.
Thank you for that wonderful introduction, and very great pleasure to be here. I would like to thank the organisers for the kind invitation to present some of our work today. I'm going to talk about optimising the EMSI test, particularly focusing on N-nitrosamine mutagenicity. So I imagine many of you are thinking, what's there to say about the EMSI test? It's been around for a long time. It was invented in the 70s by Bruce Ames, and then the assay was further developed by Aaron and Ames in the 70s. And unfortunately, Bruce passed away sort of last year, so he was a fantastic scientist. I'm not going to go into detail of how you conduct the EMSI test. I'm assuming this audience know a lot about the EMSI test anyway. But what I will say is that in industry, as applied genetic toxicologists, the EMSI test is a cornerstone for us to evaluate the potential of new drug entities for mutagenicity, and also to evaluate impurities or human-specific metabolites or degradation products so that they can be controlled and reduce any potential harm for our patients in the medicines. And more recently, so the topic of nitrosamines in drugs has become a serious issue. They were identified a few years ago in valsartan, and regulatory agencies requested pharmaceutical companies to look at their pharmacopoeias and investigate the presence of nitrosamine impurities in their drug products. And then typically, as part of that qualification, we use the EMSI test. However, there's concern about nitrosamines for two reasons. One is they're part of what we call the cohort of concern, and that reflects their potency, their potent genotoxic carcinogens, and they typically therefore fall outside of the International Conference on Harmonization regulations around how you assess the mutagenicity of genotoxic metabolites or DNA-reactive metabolites or impurities. And therefore, there's been concern also about the veracity of the EMSI test results for nitrosamines. And this is predicated typically on legacy reports in the literature suggesting the EMSI test is insensitive to nitrosamines. And therefore, if the test is a cornerstone of how we evaluate chemicals and the EMSI test is no longer accepted, it puts us in a very difficult position. You have to follow up with either mammalian cell tests in vitro or typically in vivo tests looking at mutation, and that relies on transgenic gene mutation assays like Mutamouse or Big Blue. Those are very expensive tests. There's limited supply of animals. Only one or two CROs actually conduct the test. So there's a huge bottleneck in terms of qualifying these potential impurities and coming to decisions about the quality of drugs and importantly around the safety of medicines for patients. So because of this legacy publications, where essentially they found that a number of prototypical nitrosamines were negative in the Ames test. Regulators did not really accept a negative result, whereas they were quite happy to accept a positive result, but you couldn't qualify a compound based on a negative Ames test. So we wanted to look at what factors in the Ames test assay contribute to the potency assessment of nitrosamines. And so we focused on two dialkyl nitrosamine, small molecule nitrosamines, NDMA and NDEA, and we looked at the various components of the Ames test to see which really contributed to the potency evaluation for these two nitrosamines. Prior to doing the testing, we also took samples from our rat-induced liver, which is used as part of the metabolizing system, and looked at the expression of cytochrome 2 CYP2E1, which is the enzyme involved in the activation of nitrosamines to reactive metabolites that ultimately bind to DNA and result in alkylation. And we were able to show that when you're using induced S9s, you get higher expression of 2E1 both in the rat and in the hamster. And there have been reports in the literature suggesting that hamster is a better S9 metabolizing system compared to rat for nitrosamines. So we wanted to do a head-to-head comparison. This is the paper we published on these data. I'm really pleased to be able to relay that this was selected as Paper of the Year in Mutagenesis for 2024. So I'm really proud of my co-colleagues who helped for weeks. We were able to see that basically the enhanced Ames test protocol is highly sensitive to detecting nitrosamines consistent with the GSK data. We saw very high concordance. Where there were some discrepancies, these were category 5 compounds. Sometimes you had one lab that was positive, one was negative, but they were kind of equivocal in terms of potency, and these things can be expected. Again, we confirmed that the hamster was more sensitive compared to the rat liver S9s, and we saw that DMSO is perfectly adequate as a solvent, which is important for pharmaceuticals because a lot of the molecules will be insoluble in water. And then in the last quarter of 2024, we held a meeting with HESI.
Thank you very much. Actually, very important work to make sure that everybody is protected in the future. So, do we have any questions from today?
Why is the hamster S9 and then the rat S9?
So I'm just writing the paper with Maltox. Maltox is a company that makes S9 commercially and provides it to industry for testing, both Ames testing and in vitro testing of the mammalian cells. And two things came out of that work. One is that hamster livers have higher levels of CYP protein per gram compared to rat. So there's more expression of the CYP genes in the hamster than the rat. And then the other is that certain of the P450s that are responsible for activating nitrosamines are expressed at higher levels in hamster compared to rats. So it's a combination of those two elements that drives that differential response.
Any other questions? If not, I think we'll move on to the presentation.
I request the chairpersons to present a memento and certificate to Dr. Anthony M. Lynch as a token.